Welcome everyone to the fifth and final in this current webinar series. My name is Marie Latulippe and I work with ILSI North America. This series is brought to you in collaboration with the American Society for Nutrition, and what we're doing is exhibiting the ongoing work of both the faculty and students at the University of Illinois. And all of this work is focusing on the gut-brain axis and the gut microbiome. So just the usual reminder that everyone is on mute, but please use the chat function to type in your questions throughout the webinar, and we will get to those at the end of the session. This webinar is being recorded, so barring any need to mute unpublished data, we will be publishing that on our website, so please go there. A little bit about ILSI North America. If you're not familiar with the organization, we focus on nutrition and food safety science for the benefit of the public's health. We always like to remind people that we don't lobby and we don't do any advocacy, but we focus on developing science. And the way we work is that the intellectual contributions of scientists from industry, government, and academia are joined together with pooled industry funding to address scientific issues that are of common concern across those sectors. So we just want to thank everyone for joining us today. Our audience today is reflective of that tripartite network that we have and the approach that we take to all of our work. If you'd like to learn more about the organization, please visit us on one of these platforms that you see here. And with that, I think we can just go right into our event today. And I'd like to introduce our first speaker, Dr. Sharon Donovan. Dr. Donovan is a professor in the Department of Food Science and Human Nutrition and is also a member of the Division of Nutritional Sciences and the Institute for Genomic Biology. She was actually recently named the director of the Personalized Nutrition Initiative at Illinois, and she was also a member of the 2020 Dietary Guidelines Advisory Committee. So with that, we will turn the platform over to you, Dr. Donovan.
Thank you, Marie, and thank you for the opportunity to present some of our exciting data at the University of Illinois. As was just mentioned, we have recently launched a personalized nutrition initiative at the university that I'm directing. So I want to just put in a little shameless plug. If you're interested in learning more about that, we'll be having a webinar next Tuesday, the 29th at noon. And I'll be providing an introduction, and then Dr. Hannah Holscher and Dr. Naman Khan will be presenting some of their research. So if you're interested in that, you can find out, just Google IGB Science Chat at University of Illinois. I'll be turning off my webcam. So today's presentation, I will be providing an introduction to our Strong Kids cohort and talk about some of our research, primarily in the first year of life, on how early life nutrition is impacting the gut microbiome and trajectories. And then I'll be turning it over to my colleague Naman Khan, who will be presenting his work on the intersection between obesity, health behaviors, and childhood cognitive health. And then we'll be turning over at the end to Arden McMath, who is a doctoral student supported by our USA National Needs that Dr. Khan and I are co-advising. And she'll be presenting some of her initial findings as well as her longer term goals, where we'll be looking at that intersection of the gut-brain microbiome in our Strong Kids cohort. So first I'd like to just introduce our team. And so this is a very transdisciplinary team made up of faculty in food science and human nutrition, human development and family studies, and Barbara Feast and I are the co-PIs on the project. Dr. Khan in kinesiology, Dr. Taran Garcia in College of Medicine and Extension, our wonderful support staff and visiting researchers, and plus I'd have to say an army of undergraduates who, without their help, this would not be possible. And then, of course, we have to, thank our participants who, without them, we wouldn't have any data to present today. Also, this is very much a public-private partnership in terms of the funding, and I really need to acknowledge the support of the National Dairy Council, which has supported the cohort, and then funding from NIH and USDA that's helped to support students and analyses, as well as funding through the Food and Family Program by the Family Resiliency Center with funding from the Christopher Family Foundation and also the Gerber Foundation. So to jump right in on the development of the gut microbiome, I think we've all heard over the last decade about the fact that our bodies harbor diverse, complex, and abundant microbiota. We know the composition is influenced by many factors, and as I'll show you, during early life is a particularly critical time for establishing that microbiota and getting it sort of off on the right foot. We know from preclinical and clinical evidence that the gut microbiota is essential for normal immune and metabolic, gastrointestinal, and cognitive development and function. A lot of this evidence has been from germ-free animals, so that we know in the complete absence of a microbiome that none of these systems develop normally. But we also have some clinical evidence from humans to show that dysbioses in particular times or diseases can affect these functions. And as I mentioned, really the first 1,000 days of life is a critical time. So as we see, these are just a summary of some of the early life factors. So we have maternal factors that are acting even prenatally. We have the postnatal factors, including exposure to antibiotics, early life nutrition, genetics, and the environment. And then if we look at, more of the infant components, so how that infant is delivered, either vaginally or C-section, plays a role in the initial seeding. I oftentimes talk about this as seeding and feeding. So we have then how that infant is being fed in terms of, are they breastfed or formula fed? What happens when we begin to add complementary or solid foods? And then that important transition to more of an adult type diet. And we're learning a lot more in this area. Again, a lot of the microbiome data is descriptive and associative, but we are moving towards a better understanding of mechanisms of action between the hosts and the microbes. So then jumping into the Strong Kids. So Strong Kids stands for the Synergistic Theory and Research on Nutrition and Growth Project. And this is a birth cohort that's investigating how individual biology interacts with the family environment to promote healthy eating habits and growth trajectories in young children. It's a longitudinal birth cohort of, we started with about 468 families, and we currently have funding through 7 years of age, and it was established in 2013. And the basis for the Strong Kids is what we call the 6 C's model, where we're really taking a very interdisciplinary and cross speciation in a way, looking all the way from the cell, which includes SNPs and microbiome, to the child, to the clan or the family, the community, and the country. So our transdisciplinary team is working across all of these levels, and we're particularly interested in being able to move across disciplines. So looking at how the microbiome is affecting temperament, how that's being impacted by the family chaos and things like that. So this shows the data collection. So we have 5 time points in the first year of life: 1 week, 6 weeks, 3 months, 1 month after the introduction of solid foods, 1 year, then 18 months, and then annually thereafter. And we do home visits at all of these time points, where we collect heights and weights of both the mother and the infant. We collect stool samples from the infant at all the time points, the child and the mom. We collected at 6 weeks. We collected a saliva sample from the mother and the infant at 6 weeks, and then added another sample from the child at 4 years. And so we're interested in looking at potential epigenetic changes between the 6 week and 4 year time period. We also collected a milk sample and did a 24-hour weighing to get milk intake at 6 weeks. So we have a number of biometric measures, and we also have a very extensive survey that the parents complete at all of these time points. And this is just an idea of some of the factors that are on here. And the survey ages up. So in early times, we're asking more about breastfeeding, and now we're at 5 and 6, we're looking at the transition to school and other factors. So both a combination of qualitative, quantitative, biological data. This is a description of our cohort. So our mothers, approximately 31 years of age, predominantly white, non-Hispanic, predominantly married or cohabitating. As is indicative of a college town, we have about 73% are either college grads or post-grads, and 69% employed. I think it's important to note that even in this highly educated sample, we still had about half of our moms who had a pre-pregnancy BMI that were overweight or obese. And so since one of our major interests is childhood obesity development, we do see that many of the moms are in this overweight and obese category. The infants, again, 66% firstborn, predominantly white and non-Hispanic. We had an exclusion criteria for birth less than 37 weeks or small for gestational age. So our infants were about 40 weeks and a normal birth weight and length, and about 73% vaginal delivery, 27% C-section. So for my part of the presentation, I'm going to be focusing on these interactions. So infants have a variety of ways they can be fed in the first year of life. They can be exclusively breastfed, exclusively formula fed, or mixed fed, where they're getting, a combination of both their own mother's milk and formula. We know that these different modes of feeding can impact growth. There's emerging evidence that these different modes of feeding can also impact the gut microbiome. And so we're interested in how the gut microbiome within the context of these different feeding modes is impacting growth. And then also, what are the interaction with these other environmental factors that I previously described? So this basically shows our infant feeding mode in the first year of life. So we have a high percentage of infants who start out exclusively breastfed or combination fed. We have less than 10% that start out on formula. Again, this is a self-selected longitudinal cohort, so we recruited more moms who were interested in breastfeeding. And what surprised me is that we actually maintain a pretty high level of exclusive breastfeeding through a year. Almost about 50% of those babies had never had formula or during that first year of life. So we see in this combination fed, again, this is a group where infants may be moving from exclusively breastfed to combination fed or even to formula fed. So we're able to track each individual infant over time as their feeding modes change. But this just gives you an idea of the overall cohort. Within the combination fed, the mean was actually 50% human milk, 50% formula, but there was a range from 8 to 95% human milk. And we do see about 5.9 months is the age of complementary feeding. Again, AAP recommends between 4 and 6 months. This slide shows the infant weight for length categories in the first year, so starting at 6 weeks out to a year, and we see underweight in blue, normal weight in orange, overweight and obese based on these percentile categories. And as you see that during this first year, we go from about 14.4% of the infants falling into greater than 85th percentile to up to about 37% by a year of age. And we see a big jump here between 6 and 9 months of age. And there's a lot of factors. There's the addition of solid foods at this time. We also see more infants moving into daycare outside of the home. So we're very interested in teasing out what some of these factors are. But basically what we're seeing is that in the first year within this cohort, the infants are getting heavier. And by a year, really none of the infants are categorized as underweight. So that was looking at the full cohort. And then when we broke it down by feeding mode, we have breastfed, combined fed, and formula fed, and across the different 6 weeks, 3 months, 6 months, 9 months, and a year. And these are the Z-scores. So again, 0 would be a 50th percentile. So what we noticed, though, is that if we compare, for example, the breastfed with the formula fed, although the whole cohort was getting heavier, it was really the babies who were being exclusively formula fed were sort of leading the charge, and they were jumping up in their weight-for length Z-score already at 6 months and then maintaining it. And then these combination fed infants were somewhat intermediate. And this is just through the first year. We're now then looking at how these trajectories are carrying out through the second and third year. So another way that we looked at the data was to look at growth trajectories, which gave us a nice idea of how these children were growing over that first year. And what we found is that the infants, using a semi-parametric mixed models, the infants fell into 3 categories. And surprisingly, it was about a third of the infants in each category. So we had what we call the mid-stable, and these kids kind of track along the 50th percentile, about a 0 weight for length Z-score, throughout the first year of life. We have what we call the mid-high, which start out around the 50th, but then experience this very rapid early postnatal growth and end up somewhat plateauing, but still being almost a +2 Z-scores at a year. And then we have this group here that started out a little bit smaller, -1, and then kind of slowly climbed to being close to the 50th percentile. And I'll come back to the slopes of these lines and these lines later in my presentation, but I'll also talk later about how we looked at how the microbiome differed between these 3 growth trajectories. So we also found that there were some demographic differences in the children that fell into these different trajectories. So the gray column shows the overall cohort. And relative to the overall cohort, the kids in what we call the low late, so they started low and had kind of a late surge in growth. There were fewer females, and there were a higher proportion of females in the mid-stable. When we looked at race and ethnicity, the kids in that mid-high, so the group that showed that really earlier rapid growth, there were fewer non-Hispanic whites and a higher proportion of non-Hispanic blacks in that category. And then within the low late, we also had a higher percentage of those babies that had C-section. So, I mean, these are all sort of demographic characteristics that we need to consider when we're looking not only at how diet is affecting these growth trajectories. So then we also looked at feeding mode, and this is a little bit busy, but we see breastfed in the blue, combined fed in the orange, formula fed in the gray, and then this top are infants who were already weaned to cow's milk at a year of age. And what we observed was, if you look at the mid-stable, which we might say is the best kind of growth, that more of those infants were exclusively breastfed early on. In the low late, in the mid-high, we see relative to the mid-stable that more of the infants were being exclusively formula fed at 6 months. And then when we look at this mid-high, at 12 months, had the highest amount of proportion of infants getting exclusive formula fed, and about 11% had already been weaned to cow's milk, which is not in line with the AAP recommendations to transition to cow's milk after a year, and that compared to 3 and 5% in the other groups. So the take-home message is that all 3 feeding modes were reflected in each growth trajectory, but there were some differences in the relative proportions, and these changed over time. So now we're going to shift to the microbiome, and this is sort of, you think about the bird's eye view of the microbial composition over the first year. So this is at the phyla level, it's at a fairly high level, but what you see if we look from 1 week out to a year, that we see this sort of progressive reduction in the proportion of bacteria in the phyla Proteobacteria. We see a bloom of firmicutes at 6 months, so after addition of solids, and then the Bacteroides and Verruca microbia stay relatively constant. So then when we look by structure, so this is a principal coordinate analysis of the what we call beta diversity. And so the previous slide was looking at all of the infants, but what we see when we look at the structure, we see that the breastfed infants are clustering separately from the formula fed, and we see that the formula feds tend to cluster more tightly than the other groups, and we've seen this in other studies. And then we see this is where the combination infants are falling, and what was surprising to me is that they actually cluster more closely to exclusively formula fed infants than they do the breastfed. I really thought they would be more intermediate, given that on average they were getting half human milk and half formula. Again, there's a range, so I think that's why some of these are overlapping, but it was really suggesting that, you know, any formula was shifting these babies, at least from a microbiome. Actually, we saw the same thing from the metabolomics, fecal metabolomics, that they looked much more similar to formula fed, exclusively formula fed, than exclusively breastfed. So this is going back to the looking by feeding group, breast, combined, and formula, at the phyla, genera, and species. And I didn't put any names here because I just want you to get a big picture and appreciate, again, that as we look at these two, either any formula or exclusive formula, they tend to look more similar to each other than they do the breastfed. And also, as you see, when we look at the genus and the species, more diversity, that there's more bars here and more species, and we'll come back to that later. So then we look statistically to see what were the factors that were affecting this microbiome diversity. And basically it was significant over time. So every time point overall was different. Then when we looked at the breastfed versus the formula fed, they were significantly different from each other at all time points. So even after the addition of solids, they were significantly different. When we looked at the breastfed versus the combined fed, they were different again at all time points, except this didn't reach the level of significance. And then when we looked at the formula versus the combined fed, they were only different at these the first 2 time points, the 1 week and the 6 week, and then they were no longer significantly different. So I think that this is novel data to really suggest that formula is coming in, or less breast milk, to shape the microbiome in unique ways. So that was beta diversity. This is what we call alpha diversity. So this is looking at how much diversity within a sample. And what we observe, so this is going back to our growth trajectory data. So we found that the babies in this mid-high, so again, the ones who showed that early rapid growth and ended up being at a higher Z-score, had a more diverse microbiota at 1 week, 6 weeks, and then 6 months. And oftentimes for adults, we think a more diverse microbiota is healthier and better. But if you think about it, a breastfed infant actually has a really typically the bifido-dominant microbiota. So in this case, in infancy, we oftentimes think that having a less diverse microbiota might be better. But either way, we found that a more diverse microbiota was associated with this more rapid early growth trajectory. So then we asked the question, well, what were differences in the infants over time in these trajectories? And the first column shows the phyla, genus and species, and then time. And basically what we found is that earlier, you can see this is almost all firmicutes except for the Akkermansia, which is a Verruca microbia. Earlier and more abundant colonization with firmicutes was more common in the infants, statistically more common in infants in this mid-high growth trajectory. So they were being colonized to a greater degree, even at 1 week of age, in Ruminococcus navis, which is a very common commensal microbe in adults. We typically in infants don't see it coming up until after we introduce solid foods. So it's rare to see it this early. And you almost see this progression as well. So Glaudia also, which is previously categorized as a ruminococcus, and you start to see Clostridial species being more predominant at 3 months than the Akkermansia anostipes and another ruminococcus. So in all of these cases, these are more predominant in that early rapid growth trajectory. However, this flipped. So Ruminococcus navis, all of a sudden at 6 and 12 months, was actually higher in that kind of low late group that we call. And so that caused us to go back and look at these trajectories, which I mentioned before. So if you see this low late, kind of a relatively flat trajectory, and then a greater slope, a greater trajectory. And that's sort of the opposite of what we see in the mid-high, where it's more rapid in the 6 months and flatter in the second 6 months. And so we're hypothesizing that this is why this is flipping, so that actually this Ruminococcus navis is greater in abundance during periods of more rapid growth trajectories. So the overall summary and conclusions for my section, I hope I've convinced you that early life is important and that nutrition is a primary determinant. We found identified 3 growth trajectories in this first year, and those infants in the mid-high, which again in other studies has shown that pattern of growth is a greater risk factor for obesity, childhood obesity as the kids get older. So those, they were less likely to be breastfed or combined fed versus the mid-stable. They had that more diverse microbiota at 1 week, 6 weeks, and 6 months. And they had this earlier colonization. And so we started to look then, it's like, well, what is there some, supporting evidence or some biological context? And there was, most of us in the microbiome, we know Jeff Gordon's group, and he had a postdoc, Laura Blanton, who was looking at the microbiome of kids from Malawi. So they had well-nourished kids from Malawi and undernourished kids from Malawi. And within their notobiotic mouse model, they were able to show that Ruminococcus navis and a Clostridium symbiosum, actually, when they co-housed mice with these different microbiota, that these 2 species moved from the well-nourished to the undernourished microbiota. So they basically, mice are coprophagic, so they were eating the stool, and that these 2, which were not in the undernourished microbiota, when they consumed the stool from the mice with the healthy microbiota, those moved over. And then they did another study where they actually gavaged those into the mice. And what they found is that adding these to the undernourished microbiota caused those mice to grow more rapidly, and it also corrected the metabolic abnormalities. So this is a study which, again, in malnourished children, it would suggest, well, maybe this would be beneficial. This could be a probiotic to promote growth. But our studies showing even this strong statistical association in normal healthy infants, that having a higher abundance of Ruminococcus navis is associated with more rapid growth. So I will now turn it over to my colleague, Naman Khan, for the next section of this talk.
Great, thank you, Dr. Donovan. And we would love to introduce Dr. Naiman Khan. Dr. Khan is an assistant professor and the lead of the Body Composition and Nutritional Neuroscience Laboratory at the University of Illinois. And his group examines the interactions between nutrition, abdominal adiposity, and cognitive and brain health, with the aim to eventually develop effective dietary interventions to mitigate the detrimental effects of obesity and metabolic risk. So with that, I'll turn it over to you, Dr. Khan.
Thank you so much. First, I'd like to thank ILC North America, DNS, and ASN for hosting this wonderful series. It's a great opportunity for faculty and our staff, our students, to also share their research My group is a relatively new addition to the Strong Kids team, and we're really excited about working with the group. We have historically examined the relationship between health behaviors like obesity, physical activity, and nutrition, and cognitive health in older pre-adolescent children. And with general support that we received from the NIH and NDC, the National Dairy Council, we've had an opportunity to extend this work to the younger cohort in Strong Kids. So what I'll be doing today would be to just give you a brief overview of how these health factors in older school children, but also in perhaps the 4-year-olds age range contribute to cognitive function. So for background, I was hoping to first highlight just some brief epidemiological data that I don't think is going to surprise a lot of people, but I think it's important to take a step back and sometimes think about the lifespan a bit more carefully. So these are data from the CDC, just highlighting the prevalence of obesity and the proportion of individuals meeting physical activity guidelines and those meeting fruit consumption guidelines. It's estimated that approximately 20% of children have obese weight status, and this often tracks into adulthood. Currently, about 40% of adults have obesity. We also see similar patterns of poorer activity, percentage of individuals meeting physical activity guidelines as individuals get older. And we also see this in the case of people meeting guidelines for vegetable and fruit consumption. While the influence, and I know from the literature we are all generally familiar with, there's quite a bit of literature on the interactive influence of obesity, nutrition, and physical activity on cardiovascular health. Increasingly, however, we're learning that these health factors are also important for cognitive function as well. So they, the brain and its abilities are not immune to all the cardiovascular impairment or even the risk factors associated with obesity and poor health behaviors. So hopefully by the end of this talk, I'll have shown you some data that would convince you that these factors are important even in childhood. So as far as understanding the implications of health behaviors and obesity for cognitive function, we have focused our work on a set of functions known as executive function. Executive function, which is also known as cognitive control, is a set of processes that are vital for regulation of our behavior. These include working memory, inhibitory control, and cognitive flexibility. Working memory is the ability of an individual to hold information in your mind and manipulate it. Inhibitory control is the ability to regulate attention and regulate self-behaviors. Cognitive flexibility is often called multitasking. Together, these executive functions are thought to provide support for higher order functions that we typically think about as brain function, like reasoning, problem solving, and planning. What's important to think about when it comes to executive functions is that there's been hard data collected that shows that executive function abilities predict greater long-term scholastic and vocational success, as well as more marital success, happiness, as well as financial plentifulness in later life, less risk for substance abuse in adulthood. So we know that executive function in childhood can be an important marker of these real-life outcomes in adulthood. What we also know about executive functions, and I think is important to think about in childhood, is that these are dependent, or at least disproportionately dependent on the prefrontal cortex, which is a region of the brain that exhibits a protracted development all the way until the 3rd decade of life, which allows for health behaviors to have an impact in terms of optimizing cognitive function. So as I go through the presentation today, I'll go through just the 3 different areas that we've done some research and highlight some key studies. I'll start with obesity, then physical activity and fitness, and then highlight some work and recent work we've done in the area of nutrition and childhood cognitive health. So one of the first studies we conducted in this area was when I was a graduate student here at the University of Illinois, in collaboration with Charles Hillman's laboratory. And their laboratory had done research looking at the effects of aerobic fitness and cognitive function in school-age children. My interests were focused on obesity, so we were able to actually ask some very basic questions about the effect of obesity on children's cognitive function after we have accounted for well-known factors like aerobic fitness, IQ, and socioeconomic status. So in this study, what we did is we matched students for those important covariates. We compared healthy weight participants between the age of 7 to 10 year olds, those healthy weight versus those with obesity. We compared their performance on a go-no-go task. The important thing to appreciate about this task is that there are two conditions. There's a go condition and a no-go condition. In the go condition, participants are presented with one of two stimuli on a screen, and they're asked to respond to the rare stimulus. In this case, it was the cartoon character of a lion. That occurs about only 20% of the time. So it's a basic attention task. It's easy to do. Children just essentially just wait until they see this the random lion show up. Only 20% of the time they can press the button, and then we can measure their performance. In the no-go condition, on the other hand, it's actually an inhibitory condition where we reverse the instructions. So the child then is asked to respond on a response pad for the more frequent stimulus, which is the tiger on the screen. And so most of the time, the child is actually pressing a button, and when they see the target stimulus, which is the lion, they have to withhold their response, which actually necessitates inhibitory control. So when you compare participants who are healthy weight versus those with obesity, what we learned in this study was that children with obesity actually had poor performance, especially and only evident in the inhibitory control condition. So when you actually have inhibitory demands increase, when you see this difference emerge. Additionally, we also have the capability in our laboratory to not only study behavioral performance measured in terms of accuracy and reaction time, but also at the neuroelectric level. And we do this using event-related brain potentials. These are a component of the ongoing EEG as participants do these tasks. Specifically, we're interested in examining the P3 ERP component. This is the component that typically emerges about 300 milliseconds post-stimulus onset. It's a waveform essentially with depending on its amplitude, it gives us an idea of how much attention somebody is dedicating to a particular task. When that peak actually occurs, gives us an idea of information processing speed. So that gives us, it allows us to go beyond simply looking at behavioral data and actually understanding how a child is utilizing their mental resources as they do these tasks. So in this task, when we looked at the P3 data in these participants, especially when we compared the no-go versus the go condition, we realized that the healthy weight children actually exhibit a modulation in the P3 index, but what this showed was that essentially there's a larger difference in the P3 between the go and the no-go condition in the healthy weight participants. However, we don't see that sort of modulation in children with obesity. So this was the first study that not only highlighted that there's poor inhibitory control in children with obesity, but also that they were inefficiently utilizing their neural resources as they're doing this task. However, one of the limitations of this previous work it was the fact that we relied on BMI, and it's well known that BMI, while a very useful tool in terms of weight categorization, doesn't account for many factors, including body composition and fat distribution. And we know from other studies in the area of cardiovascular health and glucose regulation that central adiposity or accumulation of body fat, particularly in the visceral cavity, predicts onset of cardiovascular disease. And interestingly, that relationship has also been demonstrated for the onset of dementia among adults. So these are data these are findings from one of the earliest studies that was done on this topic. It was a large study that was done with thousands of participants where they followed them for over 30 years, starting in their mid-30s, and they looked at their diagnosis of dementia 30 years later, based on quintiles or categories of their different anthropometric measures. So those in the quintile 5, for example, those were that had the highest BMI, highest subsatchel abdominal diameter, highest thigh circumference in their midlife. And what these data revealed was startling, was that individuals who are in the highest quintile of sagittal abdominal diameter, which is essentially the height of somebody's waist, height of their belly when they're lying horizontally, was actually predicted the onset or diagnosis of dementia up to almost a 3-fold greater risk, even after accounting for BMI. So we know that in adults, central adiposity is a predictor for dementia risk later in life. So increasingly, our work in children has focused on this hypothesis that metabolic impairment that we typically see in adults that impacts perhaps cognitive health, but also well known for its impact on cardiovascular health, may be also evident in younger children. And we have done some work in pre-adolescent children showing that. But I just wanted to highlight that we've also done this work in children as young as 4 years of age. So a couple of years ago, we had pilot funding from the National Dairy Council that allowed us to test cognitive assessment tools, and even just ask the basic question of whether the relationship between obesity and health behaviors, whether, since we've observed them in older children, whether they can be observed even in children as young as 4 years of age. So this is a recent publication that we have looking at the comparison between whole body percent fat and visceral adipose tissue, both these assessed using DEXA, comparing both these metrics across different measures of neuropsychological function in 4-year-olds, including intellectual ability, expressive language, and academic skills. And what these findings revealed is that when you actually categorize the participants based on their visceral adipose tissue, that's when you actually see differences in their expressive language and academic skills emerge. However, not when you look at their BMI percentile or their whole body percent fat. So these data essentially support what we're seeing in older children and perhaps even in adults, that visceral adiposity and central adiposity metrics are more sensitive to cognitive function. And it was interesting and perhaps even a little alarming to see these relationships in children as young as 4 years of age. So the next few slides, I was going to cover some knowledge that we have in the area of physical activity and aerobic fitness and its relationship with cognitive function in childhood. As I mentioned earlier in the presentation, there is only about half of children in pre-adolescence meet the recommended guidelines for daily moderate to vigorous physical activity. This number reduces by adolescence, and by young adulthood, only a third of individuals meet guidelines for physical activity. So several years ago, my mentor and colleague Charles Hillman conducted the first randomized control trial called FITKids to address a very simple question, which was whether providing children with the opportunity to meet those daily guidelines would have any impact on executive function. So this randomized control trial was funded by the NIH, enrolled 221 children between 7 to 9 years of age. It was a 9-month after-school physical activity program. It took place here at the University of Illinois campus, right next to our building in one of our rec centers. Participants were brought to campus. We actually had buses that facilitated this, and participants' parents picked their kids up from school, from the campus. They spent every day with us for weekdays, about 2 hours or so, and in that 2-hour window, participants engaged in moderate to vigorous physical activity and using sports, but also some low organizational games. And the measures of this study were body composition using DEXA, aerobic fitness using a VO2 max test, and then also attentional control and cognitive flexibility, among other measures. So what you're seeing on the right is a figure illustrating a comparison between the control group and a weightless control group and the treatment group. And as expected, both groups improve in executive function measures of attentional inhibition and cognitive flexibility. It's anticipated that that would be the case because that's as a function of development, and just growing older children get better at these tasks. However, what you see is that in the group that's receiving the daily physical activity intervention, we see greater benefits in both attentional inhibition and cognitive flexibility. So this was the first study that demonstrated this using a randomized control design. Since then, several other studies have come out and published in similar age cohorts, demonstrating the importance of provision of physical activity, even over short durations, even in acute settings. This has actually been the provision of physical activity and benefits for cognitive function have been adopted in the recently published physical activity guidelines for Americans. But what was interesting, and I wanted to just highlight because of our interest in obesity, the interpretation of these findings within the context of childhood obesity, I think, is very interesting. So recently, there's a publication that I had the opportunity to contribute to, where we looked at the FITKids data, secondary data, and we examined the results for the neuroelectric component, as I mentioned, the P3 earlier, and looking at the patterns of changes in the P3 as a function of baseline weight status in these participants. So what you're seeing here is a topographic plot of the P3 in healthy weight versus those children with obesity pre-post, with the weightless control on top and the physical activity intervention at the bottom. And what's interesting is that in fact, we see changes in neuroelectric patterns that are pretty similar in the groups. For example, in the physical activity intervention group, pre-post, we see patterns of increased P3 pre to post in the children that are healthy weight. Even in those in the waitlist control group who start their, at the beginning of the year, but the healthy weight, we see that progression of increase in P3 over the course of the 9 months. What's interesting though is that we don't see that progression in the participants who have obesity but are also enrolled in the control group. In fact, we don't see that benefit of P3 improving over that period of development, which you actually see in the children with obesity who are getting the physical activity intervention. They have patterns that are similar to the healthy weight children. So this was really interesting because these data reveal to us that the children with obesity in particular may benefit from physical activity provision that would allow them to keep on track as children who are healthy weight as they go through development, or those that get physical activity. As I mentioned just earlier, increasingly that there's been published work that has shown that physical activity and fitness are related to cognitive function in older school children. However, the literature on preschool age children has been limited. So as part of the pilot study that was supported by the National Dairy Council, we were interested in also examining whether the relationship between aerobic fitness and executive function can be demonstrated in children as young as 4 years of age. So in this study, we assessed cognitive flexibility using a Hearts and Flowers switch task. This task is relatively simplistic, but it can be challenging depending on the rule sets you apply. So it's a computerized task. In one condition called the Hearts task, participants are required or asked to simply press left or right on a response pad based on where they see the heart on the screen, either left or the right or the fixation cross. In the flowers condition, they are asked to exercise the opposite rule. So if they see a flower, they have to respond on the opposite side of the screen. And then there's a block of trials that we administer that actually mixes these trials. So this is a case where the participants have to recall the rule set changing, where if they see a flower, they have to switch and respond to the opposite direction. If they see a heart, they have to go back and respond to the same direction as they see the heart on the screen. So that can be a very challenging task. And what it requires essentially is children to multitask or switch rule sets as they do it. We assessed aerobic fitness, so using a submaximal test of aerobic fitness, which is a field measure called a 6-minute walking task. In this task, participants are asked to walk a fixed distance, do as many laps as they can over a 6-minute period. This is the well-validated approach of determining submaximal aerobic fitness in children. And we were able to relate these two metrics in the data here on the right. First, it was really important for us to demonstrate that this task actually can be executed, and then you see the data that you expect in terms of just the performance across the different blocks. So as you can see at the top here, the accuracy for the hearts condition, the flowers, and the mixed. As expected, the mixed trials are the hardest, where children the performance tends to suffer. That's because they have to exercise those different rules and remember which rules are applied based on the stimulus. And then when we look at the performance during those mixed blocks, called the heterogeneous accuracy, we actually see that aerobic fitness, as measured by the number of laps that children can walk, is actually positively related to accuracy during that condition when they have to apply those difficult rule sets. So these data were very interesting because they were consistent with what we had seen in older children, but they were able to also provide us with some very important tools that we could then use in future studies. In addition to examining the aerobic fitness relationship with the behavioral task, like a switch task, we were also interested in the P3. And you can in this study, we assessed the P3 using an auditory stimulus, which is an auditory oddball. And the stimulus were essentially just high-pitched and low-pitched tones. And one's called a target and one's called a standard. So the target is that high-pitched tone that occurs more rarely, and similar to what I've described earlier in terms of the go task, which is a visual stimulus. This is an auditory stimulus, and it's a rare stimulus. So when participants actually hear that high-pitched sound, you see different neuroelectric activity at a higher P3. So I have a short video here that would just illustrate the task for you. Perfect. So hopefully if you're, I doubt, I'm sure many of you fell asleep while I was presenting, so at least now this will wake you up. But that's, as you see the child responding to the high-pitched tone, we expect a different neuroelectric waveform, a larger peak in the P3. So these results illustrate that during the targets, the participants who are higher fit tend to have a higher P3. And when you actually compare the P3 during the targets versus the standards for the high fit children, you see a bigger modulation in the P3, suggesting that their neuroelectric activity also improves or increases when they actually need to do the task at a higher level in terms of difficulty. So these data not only replicate studies in older children, both for behavioral performance as well as the neuroelectric data. So for the last few slides, I was going to highlight some work we've done in the area of nutrition and its implications for cognitive function in childhood. Specifically in the area of nutrition and cognition, we've focused our efforts examining the impact of the implications of carotenoids, specifically the carotenoid lutein. Lutein is a carotenoid or plant pigment that's found in highest quantities in green leafy vegetables, but also in small but highly bioavailable amounts in egg yolks and avocados. So you can think of like an avocado toast being a very good source in terms of bioavailability for lutein. Lutein and other carotenoids have, of course, been long studied for their as dietary bioactives, owing to their antioxidant capacity for a variety of chronic diseases and health implications. They're also thought to mark a healthier diet or higher diet quality. What we know, however, that's interesting about lutein is that even though it is only contributing to about 12% of dietary carotenoids consumed, it's not one of the higher consumed carotenoids when you consider a beta-carotene or lycopene. However, when we look at the deposition of carotenoids in human brain samples, lutein accounts for almost 60% of carotenoid content in brain. So what you're seeing on the right here is the carotenoid concentrations of different carotenoids across different cortices, and we see that lutein accumulates almost 2 to 3-fold greater amounts, regardless of which brain region you're studying. So this is really intriguing data because it tells us that that lutein is preferentially being accumulated in brain tissue, even though it's not a big feature of our diet. And so there's been an increasing amount of research now really investigating the implications of lutein for cognitive function. We've done a few studies in the area of lutein and cognitive function in childhood and also adults, but I just want to quickly highlight a study keeping with the theme of executive function and the P3. So we did a cross-sectional study a little while back looking at the performance of children who have higher macular pigment optical density, which is a non-invasive proxy measure of lutein accumulation in the eye, in the macula. And it's been shown to be a validated, reliable way to measure lutein that may also be in the brain. So it's a nice non-invasive way to do this. And we were able to look, what we did in this study is examine children's performance on an attentional inhibition task and across different levels of macular pigment optical density, or MPOD. And what we observed is that participants with higher MPOD tended to do better on the incongruent task version of this task, which is a more difficult task where we actually elicit more interference control in these participants. When we look at the P3 data that we collected while participants are doing this task, it is very interesting that participants actually exhibited, the higher MPOD exhibited lower resource allocation, which is a lower P3, while being more accurate, which suggested to us that they actually might be, that lutein is actually related to neural efficiency during this task. Subsequent to that, we conducted a randomized control trial that examined lutein and DHA effects. And this is based on some adult studies that have been done that have showed that lutein and DHA, when provided together, improve cognitive function in older women. So this was one of the basis of the design for children. So in this study, what we did is we provided children between 7 to 9 years of age with lutein and DHA through a fortified beverage over a 9-month period. On a daily basis relative to a placebo. And we measured changes in macular pigment optical density as well as some cognitive tasks. And what we learned was that macular pigment optical density can improve following the consumption of lutein, even at a small dose as 1 milligram. And we did see improvements in relational memory and attentional control in the intervention group to a greater extent than the control group following the 9-month period. So for the last 2 slides, I just wanted to highlight that we're really excited about the possibility of working with the Strong Kids cohort. It's very important to recognize that much of the work I've presented today has a lot of limitations, especially the fact that we're not accounting for important early life factors that can contribute to childhood health. For example, brain development occurs at a very rapid rate early in life, and by 2 years we have almost 80% of the weight of the brain already achieved. So there's a lot that happens in the first 2 years of life. We also know that a lot of different processes essentially shape how optimal cognitive function early in life. So with the Strong Kids group, participants will have data, and Arden McMath will share discuss some of this, but we have some cognitive data in the earlier stage, so 2 years of age. And also we know that the trajectory of growth for these different cognitive processes varies in participants in children as they get older. So we'll get an opportunity to understand the longitudinal growth of these processes and the extent to which the factors I discussed today, like lutein, weight status, and body composition, may have implications. In the case of lutein, what we know is that it's more bioavailable in breast milk, and we know that infants who receive breast milk or formula with lutein in it tend to have greater serum lutein status. So we know that these factors are important to consider when we're interpreting even relationships later in childhood. So the summary slide or the conclusion for my presentation, hopefully the data have convinced you that obesity may have negative implications for cognitive control or executive function in children, pre-adolescents, as well as 4-year-olds. We see that there's a consistency there, and we can see that these relationships emerge even earlier now. We're looking forward to doing the work with the Strong Kids cohort because it has a potential to address really important questions about growth trajectory, the changes in nutrition and early feeding, but also it allows us to look at cognitive function within the 6 Cs model. So that's really something that hasn't been done in literature, is looking at how health behaviors impact cognitive function while considering factors like genetics, like microbiome, also family, and beyond that. So this is going to be a really great opportunity to really expand the pie when it comes to explaining the variability in cognitive function using health behaviors. Thank you.
Great.
So with that, go ahead.
Oh, no, I was just going to introduce the next speaker, but maybe have a couple more comments.
Oh, no, I'm okay. Thank you.
Okay, great. Thanks, Dr. Khan. Then to introduce our final speaker, that is Arden McMath. And Arden is beginning the second year of her doctoral studies in nutritional sciences this fall. She previously attended Miami University of Ohio, where she earned her BS in dietetics and then her MS in exercise and health science. She is co-advised by Drs. Donovan and Khan, and we welcome Arden today, and we look forward to hearing about your work.
Thank you, Marie. So as she mentioned, and Dr. Khan mentioned, I will be presenting on the aims and preliminary analyses of relationships between anthropometrics, health behaviors, cognitive control, and the gut microbiota in preschool age children. So briefly, just to cover what I will be talking about today, first I'm going to go through what we know already about these relationships between weight status, health behaviors, cognitive control, and the gut microbiota. And then I'm going to go through how this led me to some questions and some aims that I have for the Strong Kids 2 cohort. And then I will cover some preliminary findings of some analyses I've done in the 2-year-olds. And then finally, I'll wrap things up with some future directions. So just to briefly sum up what we've learned so far from Drs. Donovan and Khan, we know that weight status, diet, and physical activity have implications for cognitive control. We also know that early life diet has roles in modulating gut microbial colonization and function. Aside from the work presented by Drs. Donovan and Khan, though, evidence also suggests that the gut microbiome may have an influence over health in the long term. Taking, for example, the study by Stanislawski and colleagues who showed that the gut microbiome at 2 years old explained 50% of the variance in BMI at age 12. So this may tell us that the gut microbiome may be important for identifying children at risk of overweight or obesity, and may be important for subsequent prevention efforts. And while the explorations of the gut microbiome relies heavily on work in animal models and adults, there have been several studies that explore this in children. Specifically, Carlson and colleagues were among the first to explore these relationships in a longitudinal analyses. They found that gut microbiota composition at age 1 was associated with cognitive function at age 2. And specifically, they found that Bacteroides species were associated with higher scores in the areas of language development, IQ, and inhibitory control. And this study is important because it provides evidence for the gut microbiome's possible role in cognitive function trajectories. Another important study that I wanted to cover, published earlier this year, evaluated behaviors associated with cognitive control and executive function. And what they found is that there were relationships between specific taxa. So as I was saying, another interesting study published earlier this year found that these behaviors that were associated with executive function were correlated with specific taxa and functional genes that have already been linked to cognitive health. And also what was interesting about both of these studies is that they found that the species Bacteroides fragilis exhibited the strongest associations with behavior and cognitive function. And Bacteroides fragilis is known to be have some protective roles against pathogen-associated inflammation, which may be the mechanism by which this occurs. And then the last thing I just wanted to mention is that that second study by Flannery also showed various associations between butyrate-producing bacteria and behaviors with mixed results. So in animal studies, butyrate is pretty widely regarded as having a positive effect on cognitive function. However, in this study, 2 of the 3 butyrate producers were associated with poor behavior outcomes. So therefore, more explorations on the effects of butyrate on cognitive function, especially in children, are needed. So while explorations in the gut-brain axis, myself. So with the background in mind and using the longitudinal strength of our Strong Kids study, these are some of the questions that I hope to address with the Strong Kids 2 cohort. And I believe that these are particularly important because they may be indicative of early intervention for preventing childhood obesity and resulting cognitive deficits. And it will also allow us to understand if there's a particularly crucial time that children are more susceptible to cognitive deficits that are dependent on modifiable behaviors. And specifically, I think that the 24-month age is especially interesting because this is when the child has substantial developments in neurocognitive function and the gut microbiome. Not to mention, they're also forming independence from their caregivers, so they're developing habits of their own. So in order to answer these questions I have about the cohort, these are some of the measures that I will be using for the 2-year-olds. So I'm going to be using the weight for length Z-scores as recommended for 0 to 2 years. I will be using the behavioral rating inventory of executive function, also known as the brief P, to assess cognitive function. And I'll go into more details on that here in a minute. And for diet, we looked at their intake with a food frequency questionnaire that asked about their intake within the last 6 months. For physical activity and fitness, and well, for physical activity and sedentary activities, the surveys used for this were previously validated and specifically asked about the number of activities performed in a week. And also the Common Sense Media Survey for media asked about the amount of time spent using various screen devices, including video games and watching TV, DVDs, shows on various devices and things like that. And then last but not least, the gut microbial composition and function will be assessed at all time points using the high throughput sequencing and the metabolite analysis. So as I mentioned, I would explain how we're assessing cognitive function at this age. And while it would have been great to be able to do some of the tasks that Dr. Khan mentioned. Unfortunately, these are not possible in children this young, so we resorted to this validated brief P survey. And essentially this survey asks about everyday behaviors that are associated with executive function in home and preschool environments. And these questions are, as you can see on the slide, just about everyday behaviors. So how often are they overacting to small problems? Never, sometimes, or often. So in other words, in this case, a higher score indicates that the parent was reporting more behavioral issues. And these questions were all summed up to come up with these subscales that you can see at the top that aren't highlighted. So the inhibit, shift, emotional control, working memory, planning, and organizing. And these are all various aspects of executive function. And then from those, the indices for executive function were computed. So inhibitory, self-control, just as a review of what Dr. Khan mentioned, is essentially their ability to modulate responses, behaviors, and emotions, whereas the flexibility index is their ability to adapt to responses or actions according to their environment. And then the emergent metacognition index is their ability to systematically problem solve. And then finally, the global executive composite just includes all three of these indices and is based on their overall executive function. So in order to evaluate how their executive function was relating to various health behaviors, I wanted to look at their adherence to various recommendations, and specifically I chose the Bright Futures initiative to for the recommendations, and I chose this because it is supported by the American Academy of Pediatrics, and it's very recent and is continually updated based off the evidence, and it's widely used by clinicians and families. And when you Google search it, it's one of the first things that comes up with resources for recommendations for these aspects of health behavior. And then additionally, I also felt that it was a good representation of the 4 ecologies proposed to interact to affect child dietary and health habits and weight gain, as noted in the 6 C's model. So they talk about aspects of cell, child, clan, and community, and that's all encompassed in these recommendations. So the recommendations specifically that I chose to use were whether or not a child consumed at least 5 servings of fruits and vegetables, whether or not they limited their their screen time to less than 1 hour a day, whether or not they eliminated their intake of sugar sweetened beverages. And then finally, I used the recommendation that these children would be physically active every day. And I did adopt this sort of from another initiative, but also this was kind of the more broad recommendation given by Bright Futures, just due to the nature of the survey that we used to assess physical activity, because it was asking about the number of activities rather than the time. So just to give you a brief overview of who our sample was at 2 years, just of note, most of the children are of healthy weight, with only 11% having overweight or obesity. And then I also wanted to point out that just as with the overall sample that Dr. Donovan presented, there is a disproportional amount of high socioeconomic participants in the sample, with 50% of the moms reporting their highest level of education as having a postgraduate degree. And household income was also factored into this score. And then to further describe our sample, the average time spent using screens exceeded the recommendation, with 55% meeting recommendations for less than 1 hour per day. And of note, only 14% of the parents reported that these children were not being exposed to screens at this point. which is kind of interesting considering that the AAP recommends no exposure to screens before 18 months. And the number of activities reported in a week was about 10.5. And most kids, so around 75%, were meeting the recommendations to be physically active every day of the week. Additionally, going along with that theme, most participants were adhering to the recommendation to eliminate sugar-sweetened beverages. And then finally, kind of opposite to that, the fruit and vegetable intake actually had very low adherence, with only around 4% of the participants consuming 5 servings per day. So the first question that I wanted to ask is, at what ages are fruit, vegetable, and sugar-sweetened beverages and physical activity habits linked to childhood weight status? And in order to assess this, as well as my other outcomes, I did use multiple linear regression, and the covariates in this were sex, socioeconomic status, ethnicity, and birth weight for length Z-scores. And the hypothesis for this question was obesity-related child behaviors, so diet, physical activity, and sedentary behaviors would be associated with weight status at 2 years of age. And what we found is that only screen time explained any of the variability in weight status at 2 years of all of the variables. So as they had higher screen time, it was associated with a higher weight for length Z-score. And interestingly, birth weight for length Z-score did not explain the variance at 2 years old, but it was correlated. So the second question I had was, when do health behaviors and weight status begin to show relationships with cognitive function? And the hypothesis being that weight for length Z-score would be positively associated with and predictive of cognitive function at 2 years of age. And what we actually found is that the 2-year weight for length Z-score did not explain any of the variance in executive function at this time. My next hypothesis was that relationships between obesity-related behaviors, so diet, physical activity, and screen time, and cognition would exist at 2 years of age. So in other words, I wanted to know, since the relationships between weight status were not related to cognitive function quite yet at this age, is it simply that maybe some of the health behaviors that could perhaps ultimately lead to overweight or obesity, maybe these have relationships before weight status actually does. So what we found is that diet actually showed no significant relationships with cognitive function indices, but screen time was once again the variable with the strongest association. And as you can see, as the children spent more time using screens, their brief P-index scores increased, meaning that the parents reported a higher incidence of behavioral problems that are associated with executive function. And similarly, adherence to the recommendation to be physically active every day explained a small portion of the variance in overall executive function scores, showing that those who met the recommendation tended to have a lower incidence of behavioral problems. And finally, we showed that there were significant differences between the mean scores of those adhering and not adhering to recommendations for physical activity and screen time in that chart there on the left. So my conclusions from these analyses were essentially that more screen time was linked to higher weight for length Z-scores and a higher incidence of behaviors that are associated with poor executive function. So it did explain a small portion of the variance in weight for length Z-scores, about 2.5%. And then additionally, we saw that it explained anywhere from 3.5 to 4.5% of executive function, depending on the index. And then we also saw that physical activity exhibited relationships with overall executive function, with adherence to the recommendation being associated with a lower incidence of behaviors associated with poor executive function. So another ongoing part of the Strong Kids 2 project is my work I'm currently doing with the 4 and 5-year-olds, and I'm really excited to work with the analyses on this project, as we have a lot of objective and novel measures for this young age that Dr. Khan already pointed out to you. So that kind of brings me to my next point, that there were some limitations of my analyses in the 2-year-olds. So for example, we used anthropometrics rather than body composition, which body composition will ultimately allow us to be more precise in our analyses and our conclusions, since the metabolic functions of various tissues can be quite different. And also, as Dr. Khan mentioned, may have different relationships with cognitive function as well. So these are the objective measures that I mentioned. Just to kind of reiterate what Dr. Khan already covered, we'll be obtaining the DEXA, bioelectrical impedance, we'll be doing the ERPs, as well as the behavioral tasks, and also assessing microbiome at this age. So this brings me to my last question, which is, does the gut microbiome modulate relationships between weight status, diet, physical activity, and sedentary behavior with cognitive function? Specifically, I hope to answer questions that I have listed, such as, are the anti-inflammatory properties of Bacteroides, or maybe the butyrate or butyrate-producing bacteria, are these responsible for relationships observed with cognitive function and behavior? And the great thing about Strong Kids is I will be able to explore this both as a snapshot in time, so cross-sectionally, as well as through a longitudinal lens. And I also hope to continue my analyses that I did on the 2-year-olds in the ages 3, 4, and 5. So looking at what age health behaviors and weight status relationships with cognitive function begin to emerge. And based off the evidence present at this time, I think that these questions are really important and relevant, as it may help us to not only further understand the gut-brain axis, but it may also elucidate the mechanisms by which health behaviors and obesity can impact cognitive function. And before I take any questions, I just wanted to quickly thank LC North America, as well as my advisors, for this opportunity. I think especially in this unique time where we're not getting to go to conferences and things, this was a really valuable experience. So thank you. And with that, we'll take any questions.
Great, thank you, Arden. And we do have a few minutes for questions. So let's just start with Dr. Donovan. We had a question here asking if you had looked at the delivery mode of children, so by C-section or natural delivery, or is that tracked in your study so that you could examine that at a later time with your other data?
Yes. Well, thanks for the question. We do know route of delivery. I mentioned about 73% are vaginally delivered. We didn't find that it was a strong predictor, and that's consistent with a lot of recent studies that use 16S show that a lot of the differences in the microbiome composition kind of go away after 3 months of age. But I think that that might actually be a methodological issue, because some data where they've used metagenomics, or even recently even looked at phages, you could find in the same study where they didn't show differences in 16S at a year of age by route of delivery, they were able to show that those infants had different phages. And so I think that 16S may not be sequencing deep enough. But yes, we will continue to look at route of delivery throughout all of our analyses.
Great, thank you. And a question here for Dr. Khan. And the question is, were the data on lycopene in the brain from human samples or animals? And what is known about the effective age on carotenoid accumulation in the brain?
So the data I presented, and I should have highlighted this, were in human infants. for the neural tissue. So that figure that you saw on the right of that slide was an infant who had deceased within the first 2 years of life. So it's a horrible tragedy that we had for those families. But it was one of the first data set in human infants that actually then looked at carotenoid status. So we know now the differential accumulation of carotenoids in infants. Regarding lycopene, in that study they did look at lycopene, and it wasn't prevalent in many of the participants. It was, I think, in maybe 3 of the samples, so it wasn't included in those figures that they plotted. But it certainly does also accumulate in brain tissue, just in much smaller amounts. And I don't know what the data says in older adults. There is some variability of age that's associated with the carotenoid accumulation. We know that in infants, and at least from those data compared to geriatric tissue, there is a greater proportion of lutein in the brain relative to as the brain ages. So there's the thought that a lot of this accumulation takes place early in life.
Great, thanks. We'll just do a rotation here. Arden, a question for you. One part is, I don't know if you covered this, but in the screen time, does that also include playing computer games, or is it just watching shows or that sort of screen time?
Yeah, so I think that was a strength of this analysis actually, is we did include time spent playing video games. They asked about the child's use of even different modes for participating in these activities. They asked about, watching a show on a computer or playing a game on a computer, or, watching a show on a phone or playing a game on a phone. So I think the survey did a good job of covering all the different ways in which they could be exposed, and did include video games.
Okay. And can you speculate on what elements of screen time or screen exposure might be linked to the cognitive findings that you had, even though they weren't extremely strong?
Yes, yeah, absolutely. So I first became interested in looking at screen time, honestly, because I felt that it might be a good proxy for their sedentary behavior. So that's one idea, is that perhaps the greater the screen time, the more sedentary behavior they were participating in. Although I do realize, with the mobile phones and the kinetic gaming and things like that, that might be a little bit of a muddied conclusion. But yeah, I would say it could be a proxy for sedentary behavior, but also I guess I would also be remiss if I didn't mention that the greater amounts of screen time could also be sort of like a coping mechanism for parents to use on kids that were maybe a little had some more behavioral issues. But I'd say the main one is sedentary behavior. So I would like to delve more into that and get more specific with sedentary behavior as opposed to physical activity, which we will be able to do in the 4 and 5-year-olds, because we'll have accelerometry data. So we will have that sedentary behavior information.
Great, thank you. And Dr. Donovan, there's just a question here about pasteurized donor human milk. And I don't know if you know if that's been studied with respect to some of the things you are looking at, or do you have any hypotheses about that?
Yeah, so obviously in our cohort, these were all term infants that were breastfed by their mothers. I mean, donor human milk, and particularly pasteurized, would be predominantly used in preterm infants. And this is a really good question, and I'd have to say I should know this, but I'm not familiar with data that shows that infant preterms grow differently if they're fed pasteurized donor human milk versus mom's own milk. So we didn't study that in strong kids, and I'm not familiar with the data for preterms.
Okay, thank you. And also for Dr. Donovan, can you comment on what elements of human milk might be responsible for the differences seen?
Yeah, there's data from Berkoletsko's group that did some studies in Europe where they looked at the protein content, for example, of infant formula. The issue with that study is that the protein content that they looked at was level in formula versus a much higher level. But either way, people have speculated that protein may be playing a role. Another aspect is potentially the human milk oligosaccharides. There's been a series of studies now that have come out, all pretty much associations that have associated differences in a mom's human milk oligosaccharide composition with growth and risk of childhood obesity. So that deserves future study. Although I will say the studies where they've actually added HMOs to infant formula show no differences in growth of those infants. So I think right now the work with that is associations. In terms of fat content, most of that data suggests that with higher fat content that infants actually self-regulate. And if milk has more fat and more calories, they actually voluntarily consume less formula. So infants are really good at self-regulating breast milk. But, so I think if I were to put my hands on something at this point, it might be the HMOs, but, human milk is a complex matrix and we don't fully know, how these components might be interacting.
Great, thank you. And Dr. Khan, looking at your data related to the association between adiposity and cognition, do you have any idea what the underlying mechanism might be there?
Yeah, so the literature on the mechanisms is limited, and I think because of the nature of obesity and its the mechanisms that are implicated with obesity and even other diseases is oftentimes complex, and it's not probably one aspect. It's probably not going to be straightforward. Based on animal studies, we know that obesity is related to neuroinflammation, which directly impacts brain structure and function. There's also literature on obesity's impact on brain-derived neurotrophic factor and other neurochemicals that are involved in brain development. So there's that potential mechanism. It's also not clear whether the effects of obesity are really more driven by the health behaviors like physical activity and nutrition that are really driving those effects. And I think increasingly we're learning that, at least from animal models, that saturated fatty acids and a westernized diet pattern can be problematic for memory function, at least in preclinical models. In humans, it's really complex. Certainly, it's thought that obesity may impact the normal aging process, and by impacting then in terms of just oxidative stress in the brain over time. So in older adults, that's a possible mechanism. In children, however, it's probably more complex because there are also psychosocial factors. So children who are even if obesity may have some metabolic implications, we know that children who are perceived to have overweight are often subject to teasing and isolation, and there are all these other factors could also impact that. Stress and anxiety and isolation could also impact their cognitive function and some of their achievement scores. So it's a good chance that this is actually a very complicated mechanism, and I think it's also important to be very sensitive to this topic in general because we know that the link is probably not very strong. Obviously, if there was a strong link between obesity and cognitive function, given that almost 70% of adults in the United States now have elevated BMI, that would be a really, really serious issue. It's what is, I think, a better way to think about obesity and cognitive health is to think about it much like a risk factor in any other domain. When you think about obesity and cardiovascular health risk, for example, where it's not the case that everybody with obesity is going to have impaired cognitive function, but for individuals who do go on to have some cognitive impairment, obesity is part of that pathophysiology.
Great, thank you. I think we're actually at the end of our time today. So we really wanted to thank Drs. Donovan, Dr. Khan, and Arden, as well as all of the University of Illinois speakers that we've had over the course of the series for talking with us. And we just want to remind everyone to turn the slide, check out our website for future events. We've got some things going on at FITKids and IAFP. And we also have a quite significant video library on all different kinds of nutrition and food safety topics. And we welcome you to check those out. So thanks everyone. Thanks again to our speakers. And we hope you have a great afternoon and weekend.
Thank you.
Thank you.